Hello, ladies and gentlemen. For this presentation, we're going to talk about the process of transcription. Or, another way to look at it is, what we're going to do is we're going to talk about how it is that all living things use their DNA. And the first step in using your DNA is known as transcription. If you think about the word transcription, it obviously comes from this idea of transcribing. And transcribing generally means to record or to write down. So this process is all about taking the information that's in your DNA and sort of extracting it without really messing with the DNA much, because preserving your DNA is going to be a big theme in this. So the first thing to really get at is to understand that the traits that you possess, hair color, eye color, whether your hair is straight or curly, your immune system, your heart beating, like all of your physical and biochemical traits are caused by the proteins in your body. And so if your traits come from your proteins, then where you get your proteins determines your traits. So what that does is that creates what we call the central dogma of biology, which is this idea that your DNA holds the information, all right? So what that means is that the information that will give you your traits is found inside your DNA. And in all living systems, that DNA becomes sort of a version of itself in RNA, and then that RNA is used to create your proteins. And so DNA has the information for your traits. What that means is that DNA has the information for your proteins. And what we're going to start talking about today is how you get from that information to your proteins or your traits. And that's going to involve this middleman of RNA. And the first use of RNA is transcription. And this whole deal with transcription is actually taking this information in the DNA and writing it down. Remember, part of the reason that living things use DNA is because DNA is very stable. DNA doesn't really break. DNA lasts for a very long time. And so part of the process of using DNA has to involve keeping it safe. And so rather than use DNA to directly generate your proteins, in an effort to keep it safe, DNA is used by having us make an RNA copy. All right. So the first part of using your DNA is to make an RNA copy. And then that RNA copy is going to be used to generate your traits. So a couple of players in this process. First is an enzyme known as RNA polymerase. All right. Now, RNA polymerase, unfortunately, sounds a lot like DNA polymerase. It makes sense. They're both doing the same thing. They're creating polymers using DNA nucleotides or RNA nucleotides. RNA polymerase is an enzyme that creates complementary RNA strands from a DNA template. Now, again, remember, this term right here, complementary, is a very important thing, because what that means is that if I have an A on my DNA, I'm going to be putting a U on my complementary RNA strand. If I have a C on my DNA, I'm going to be putting a G on my complementary RNA strand. So it's very important that if this is my DNA, okay, the RNA that's made from it is complementary and will look like this. Now, I want you to realize that I didn't put any T's in RNA. So when we introduced this nucleic acid structures, we talked about the nucleotide uracil. And uracil is in RNA. Thymine is not. So whenever we're dealing with RNA, you will never see a T. Instead, you'll see a U. Whereas whenever we're dealing with DNA, you will never see a U, but you will see a T. Now, this process that we're talking about creates a specific type of RNA. There's actually 3 types of RNA, and we'll talk about the next 2 in the next presentation. And the type of RNA that is created by RNA polymerase is known as messenger RNA, or mRNA. Right? It's a single strand of RNA that is produced by transcription. So RNA polymerase makes that complementary strand of RNA from our DNA template, and that complementary strand of RNA is known as messenger RNA. A couple of key ideas that are really important about this process. First, you need to understand that transcription happens in the nucleus of the cell. All right? DNA never leaves the nucleus. You must remember that your DNA will never leave your nucleus. And that's part of our whole process of keeping it stable and safe throughout the course of your existence. All right? Your DNA does not leave your nucleus. So what that means is that when transcription is going to occur, RNA polymerase goes into the nucleus, produces the messenger RNA, and then that messenger RNA will eventually leave the nucleus. Oh, there you go. DNA. It never leaves the nucleus when being used. Okay? So let's talk a little bit about the basics of this process. Now, we're not going to get into like super complicated details here, but we do want you to understand the basics. So right here, I have DNA. All right? So the first thing that happens is RNA polymerase attaches to DNA. So this right here is my RNA polymerase. Now, there are sections of your DNA that are entirely codes that simply let RNA polymerase know where to attach. And they have names. We call them promoters or initiation regions. And that's not really key. I just need you to understand that RNA polymerase knows where to attach because sections of your DNA are where it can. The second step in this process is that the DNA opens up, okay? And that's key to understand, that this DNA will open so that the RNA polymerase can attach and work its way down. Now, this RNA polymerase, where it opens the DNA, that's known as a transcription bubble. You know, a little part of it's bubbling out. And you'll notice that the RNA polymerase only works on one side. All right? And it only works in one direction. Not to be confused with the band. And that direction is 5' to 3'. So just like DNA replication has to happen in a certain direction, and you know, we talked about how you have to sort of fake it on the lagging strand. Transcription also only happens in one direction. Okay? And what you can see right here, this yellow strand here, this is our complementary mRNA strand. And this is our template. All right? So the RNA polymerase actually sort of reads that one piece of DNA from 5' to 3', and as it moves down it, it's making a complementary copy. But the complementary copy is made of RNA nucleotides, and it just becomes this free-floating strand. And you can actually see down here that this strand will then just leave the nucleus. The RNA polymerase will go transcribe something else, and our DNA is done being used. All right, those are the basics of transcription. Next time we'll do translation.